So in this video, we're going to learn about it's all in the genes. So basically the structure of DNA, and then what is a gene, what is a chromosome, all of those things will be addressed in this lesson. So first off, the nucleus. So that's the organelle in our body or in the cell that stores all of our genetic information. So this illustration shows you the nucleus. This darker purple part is the nuclear membrane. It would have pores to let things in and out. The chromatin is kind of represented by these squigglies. It usually looks like almost like a strand of spaghetti all jumbled up, or a bunch of yarn all jumbled up. And then the inner part is the nucleolus, which means little nucleus. So it's found inside the nucleus. So DNA stands for deoxyribonucleic acid. So it's made up of two long strands called the double helix that you can see here. So you can see it's a twisted ladder shape. It's contained within the nucleus of every cell, and it controls all of your traits, such as your hair color, your eye color. It actually is basically the instructions for building all sorts of proteins, and those proteins will do things in your body or in your cells to make you you. And it gives orders to the cells to tell them what to do. We sometimes call the nucleus the control center, and it's because it's containing all of the DNA or the instructions for building a new organism. This is just a different representation of DNA. So this is the DNA before it's twisted. And you can see here, and we'll talk more about this a little bit later, but you can see that it's a double-stranded molecule. So there's these letters. So these are called nitrogenous bases. There's T's and A's. So T's always pair with A. And then there's C's and G's. C's always pair with G, and vice versa. So that's this double-stranded molecule, and then it twists to form that helix shape, and then it even condenses more, so it wraps around these stabilizing proteins called histones. That's beyond what you need to know. And then it just keeps coiling and coiling and coiling upon itself until you end up with this like tightly wound, visible structure called our DNA. So a couple of terms that you should be aware of. Chromosomes are the thread-like structures of DNA, and each chromosome contains many genes, sometimes thousands of genes per chromosome. And as humans, we have 46 chromosomes in each of our body cells. So there is an exception. Our gametes have half of the chromosomes. So our egg and our sperm cells would have half of the chromosomes, which you'll spend time talking about later. Genes are a portion of the DNA strand, so a portion of a chromosome that codes for a specific protein. So again, a gene is a portion of the DNA that codes for a protein. So that protein could be involved in determining your eye color, or it could be involved in determining your hair, and so on. So we're going to take a minute to talk about Roslyn Franklin. She was a scientist that performed X-ray crystallography. So she took pictures of molecules, which are very tiny, and one of her pictures was able to be used to help us figure out the structure of DNA. Unfortunately, because of the time that she was living in, she didn't get the credit that she deserved. In fact, her data was stolen from her by her lab partner and given to Watson and Crick, who helped actually finally publish the structure of DNA. But she was not given the credit that she deserved, because without her photos and without her work, we probably wouldn't have discovered DNA as quickly. So this is the famous picture that she took, and this is how scientists didn't know what DNA looked like. They knew it was the genetic material, but they were trying to figure out the structure. So in this picture, it's showing the double helix view. So this is a head-on picture. And as I said before, this photograph was pivotal in figuring out that DNA was a double helix. And once Watson and Crick got their hands on this diagram, it was very quick for them to figure out the molecular structure of DNA. So again, because of that, their knowledge, they went on to win the Nobel Prize for their work on the structure of DNA. along with Rosalind's lab partner who gave the picture to them, named Maurice Wilkins. But the question is, what about Roslyn Franklin? She did not get honored in that Nobel Prize, partly because she had already died. So you cannot win the Nobel Prize posthumously, so she was not able to be recognized. But the question is, would she have even been recognized if she had been alive? And that is a matter for debate, because some of the things that Watson said about her in his book called DNA, the Double Helix, were not very kind. So this picture shows Watson and Crick demonstrating their model of the structure of DNA shortly after their publication of the discovery. So, what is the structure of DNA? So DNA has 4 base pairs: adenine, that always pairs with thymine, so A and T, and then guanine, which always pairs with cytosine, G and C. Its structure is a double helix, and that structure is also arranged into chromosomes. And genes are sections of DNA that code for a specific protein, as we mentioned before. So DNA screening is a lab procedure that we use to test somebody's DNA for genes. Sometimes we're looking for specific genetic diseases, like PKU, or detecting certain genes that can be precursors to cancer. Sometimes the question is, should this be done? Is DNA screening an ethical procedure? So there's a lot of debate about that. So transgenic organisms are organisms that have genetic information that's been altered with the insertion of genes from another species. So genetically modified foods, pest-resistant genes in crops, so we have better yield of food. The insulin gene, so the human insulin gene, has been inserted in bacteria so that people with diabetes can now so the bacteria basically produce insulin, and we can extract that, and it's human insulin. Before this was done, people with diabetes would have to take insulin that was extracted from the pancreas of pigs. So human insulin, much better, but it's being produced by bacteria. So one of a major medical treatment for people with diabetes was thanks to transgenic organisms. So cloning is creating a genetically identical organism. It doesn't mean that the organism will be the same age, because you could clone an adult and you would end up with a baby of that adult that you cloned. They may not be behaviorally the same. They can have different personality traits, all sorts of things. Just genetically, they would be identical. So Dolly the sheep is an example of the first mammal that was cloned from adult cells, so not from another embryo. So she was born on July the 5th, 1996 in Scotland, and she died not that long after her birth, so she still was quite young, in February of 2003. So the way the cloning happened was Dolly's mother, or who Dolly was cloned from. So a nucleus was taken from a mammary gland cell, from an adult sheep, and an egg was taken out of another adult sheep. The nucleus was removed from the egg, and then the nucleus from the donor animal that was being cloned was put into the egg, and then the egg was able to divide a bunch of times now that it had the full genetic information, because it was from an adult cell, so it had both copies of the DNA. And then that embryo was placed into another sheep to act as the surrogate mother, and she gave birth to Dolly in 1996. Unfortunately, because the cells were taken from adult cells, they had already aged. So the telomeres, which are the ends of chromosomes, had already broken off considerably. So every time a cell divides, a bit of the telomeres get broken off, and once that ends, the cells don't have the ability to divide again, and the cells age. So basically, Dolly was suffering from symptoms of old age as a young sheep. So she had arthritis and other issues that you would have when you were older, even though she wasn't an old sheep. It was because the cells were taken from an adult donor. So mutations are a change in the DNA of an organism. So sometimes that can be caused by UV radiation, like this picture is showing. So a photon of UV light hits the DNA and it causes some realteration of the chromosome, or the base pairs in the DNA. So this should be reviewed, but we're going to go over the levels of organization. So we start with the cell, the basic unit of life. Cells organize themselves into tissues, which organize themselves into organs, which organize themselves into organ systems. And finally, all of those things working together makes an organism. So again, the cell is the smallest unit of life. So here's a picture of an animal cell. We can tell it's an animal cell because it has a rounded shape, and it doesn't have a cell wall, and it doesn't have chloroplasts, and the vacuoles are small. Cells will group together into tissues. So tissues are groups of cells that work together. So your skin, your muscle, connective tissue, your nervous tissue, and so on. Organs are specialized tissues that perform a specific job. So your heart or your liver or your stomach or your pancreas are all examples of organs. Organ systems are groups of organs that work together. So your digestive system or your circulatory system, they all work together. So our digestive system is made up of, this is a picture of it, your esophagus, your stomach, your small intestines, and your large intestines, and then also the other organs that are accessories. So the gallbladder and the liver and your pancreas are all parts of your digestive system. And then all of those organ systems, work together to form an organism, a living being, such as a human or an animal or a plant. And that is the levels of organization.